Friends, I am Dr. K.D. Mishra and today we will discuss white blood cells, that is leukocytes. Our body defense is equipped with a system of immunity with a special defense system to combat with infections and toxic agents. This system is composed of leukocytes and tissue cells such as mast cells. These cells defend the body from diseases in two ways. The leukocytes, that is white blood cells, plays a very important role in body defense system. Now the leukocytes plays their role in defense mechanism in immunity in two ways. One is by acting as a phagocytes. They engulf the foreign material, the foreign pathogens, and then digest them. By this way, they eliminate the pathogens from our body. Now, second one is by forming antibodies and sensitized lymphocytes which destroy bacteria and other pathogens. The leukocytes have a very important role in providing humoral antibodies, humoral immunity to us, thereby producing the antibodies. These antibodies inactivate or destroy the pathogens. Now, the leucopoiesis. Leucopoiesis is a phenomena of production of white blood cells. Apart from the stem cells which produces erythrocytes, there are other cells which are pluripotent, which are called myelocyte and lymphocyte lineage. Now these develop into colony forming units. One is the colony forming unit spleen, that is called CFUS, and another is the colony forming unit granulocyte monocyte, that is CFUGM. The third one is the lymphocyte stem cells, which is called LSC, is another lineage which is responsible for production of lymphocytes. Now the colony forming unit GM, that is G for granulocyte and M for monocyte, the CFUG produces granulocyte, that is granular lymphocytes, whereas CFUM produces monocytes only. Now CFUG further differentiates into three types of granulocytes, which are neutrophils, eosinophils, and basophils. These are named so according to their staining properties. The neutrophil takes the stain from the neutral dyes, whereas the eosinophil, eosinophil is acidic stain. So those leukocytes which take acidic stain are called eosinophils. And the third category is of those leukocytes which take basic stains. So therefore they are called as basophil. The third series is of CFUM, that is monocytic series, which differentiates into monocytes. Now here you can see a diagrammatic presentation, a flow chart of formation of the leukocytes. Now, the pluripotent cells which are present in the bone marrow, which are responsible for providing the stem cells, they, under the influence of cytokines, they produce committed stem cells. Now, these committed stem cells can be broadly categorized into two. One is white blood cell series and another is megakaryoblast. So, under the influence of leucopoietin, a hormone which stimulates the production of WBCs, white blood cells are produced in two series. CFUM, that is monocyte series, produces monoblast and monocytes, and CFUGM, that is granulocyte monocyte series, produces myeloblast, which further differentiates into neutrophils, eosinophils and basophils. Then there is another series of megakaryoblast cells. These are responsible for production of the platelets. So, under the influence of another hormone called thrombopoietin, because as you know, previously platelets were called thrombocytes. So, anything related to platelets is added with the word thrombo. So, this thrombopoietin stimulates megakaryoblast cells, which matures into megakaryocytes, and megakaryocytes finally produces platelets. Now, the production of granulocytes occurs in bone marrow. All the three, that is neutrophils, eosinophils and basophils, are produced in bone marrow. The monocytes are also produced in bone marrow. All the WBCs are produced in bone marrow and they are released in the circulating blood as and when required. Because you know, when there is an infection of bacteria, virus or any other pathogen, even parasitic infection, the role of these leukocytes comes, and therefore when such a condition arises, release of more amount of WBCs occur from the bone marrow. Now, coming on to lymphocytes. Lymphocytes are produced in lymphoid tissue. We may call it lymphoid system, which includes lymph glands, spleen, tonsils, thymus, tonsils, Peyer's patches, especially in the large intestine, then bone marrow. All these lymphocytes and plasma cells are produced in these lymphoid organs, and these lymphoid organ stores these lymphocytes. And whenever there is an infection, these lymphocytes are released from the lymphoid organs to the site of infection, where they provide defense against the invader organism, and then they sometimes returns back to the lymphoid organs. Now, coming on to the megakaryocytes, megakaryocytes are also produced in bone marrow. Then they are converted or fragmented into platelets in the bone marrow itself, and finally they are released in the blood circulation. Now, here an important factor about the leukocytes or white blood cells is their shorter lifespan as compared to RBC, which are functional for about 4 months. that is 120 days, the total lifespan of WBCs is just few hours to few weeks or sometimes few months. As we take in case of granulocytes, the lifespan of granulocyte is about only 4 to 8 hours in the circulating fluid, but they can remain alive and functional in the tissues for about 4 to 5 days. So they can live longer in the tissue as compared to the blood. Now, the lifespan of monocytes is little more than granulocytes. It is about 10 to 20 hours in blood, but after reaching in the tissue, they become larger and macrophage. So here they performed phagocytic activity to save from different disease-causing organisms by engulfing them and by digesting them. In the tissue, these macrophages can live for months, even more, until they complete their phagocytic function and then they are destroyed. Now the lifespan of lymphocytes, it may be few weeks, few months, or sometimes even more than that, depending upon the need of the body. They are continuously produced in the lymphoid organs and released into the blood circulation, and the tissue where infection occurs, then they can return back to the lymphoid system. The lifespan of platelets is about 10 days. Now coming on to a very important parameter of blood investigation is the total WBC count, because the total WBC count gives an idea about the condition of the body, about any kind of infection in the body, any kind of ailment in the body. So an elementary idea can be reached upon by counting total WBCs. In a normal person, the average WBC count is about 6,000 to 8,000 per cubic millimeter. But depending upon the physiological condition and infection in the body, if there is some sort of infection, the count of WBC temporarily increases, then it is brought back to the normal. So the normal range is considered from 4,000 to 11,000 per cubic millimeter. Now the average ratio of WBC to RBC is 1:700. That is 1 WBC is to 700 RBC. Their number is that. The number of RBC is always less, the number of RBC is always more, the number of WBC is always less. But the production of RBC is always more as compared to RBC. The production of WBC is 3 times more than the production of RBC, because that the lifespan of RBC is very short and the lifespan of RBC is very long. It is 120 days and the lifespan of WBC is very short. Now, in case of newborn, the total WBC count is high. It is about 20,000 per cubic millimeter. In full-term pregnancy also, WBC count increases to 17,000 per cubic millimeter. Now, WBCs or leukocytes are classified into two categories based on their staining properties. Those with granular cytoplasm are called granulocytes, and those without granules with homogeneous cytoplasm are called agranular leukocytes. Now, the granular leukocytes are categorized on the basis of, again, their staining properties, whether they are stained with neutral dyes or with acidic dyes or with basic dyes. Now, those which are stained with neutral dyes are called neutrophils. Those which are stained with acidic dye like eosin, they are called eosinophils, and those which are stained with basic dyes are called basophils. Now, coming on to the neutrophils, these are the most abundant leukocytes in our blood. Their identification features are their cytoplasm is granular, it is stained with the two stains called Leishman's stains and Giesma stain. These grains of the cytoplasm take stain from these dyes. They are most numerous in our blood, forming about 50 to 70% of WBCs, and their average size is 11 to 14 μm. So, neutrophils are more in number as compared to other granular leukocytes. Now, here you can see the structure of neutrophil. The neutrophil is usually having a multi-lobed nucleus. The number of lobes varies from 2 to 6. The number of lobes increases with the degree of maturity. That means as the neutrophil becomes mature and older, the number of lobes in the nucleus increases, and therefore their structure is variable at different stages, and therefore only they are called as polymorph. Poly means more and morph means shape. You know, here you can see a neutrophil with the granules stained with neutral dye, having a 3-lobed nucleus 1, 2 and 3, and this is a bar body containing sex chromosome. Now, here you can see that the neutrophils are the most active cells. They are more active than RBC. They contain a variety of enzymes inside. Their count increases in certain diseases. In case of pregnancy, when the mother is feeding milk to her child, that is during lactation period, their count also increases. After different medical conditions like hemorrhage, after burns, in pyogenic infections, that means the infections in which there is a formation of pus. Now, the increase in neutrophil count is termed as Neutrophilia. So, this word philia is repeatedly used for indicating more production of a type of cell, whereas the another term that is called as penia is used for indicating less production of a particular type of cell. So, neutropenia occurs when the number of neutrophil decreases. It is seen in certain pathological conditions, like in case of typhoid, in case of various viral infections, in case of bone marrow suppression. If some drugs are being given which suppresses the production of blood cells in the bone marrow and other organs, then obviously there will be a decrease in the count of neutrophils. We call it neutropenia. Now here again the types of neutrophils. Arnott 1904 classified neutrophils on the basis of number of lobes. As I just mentioned, as the neutrophil matures, number of lobes in the nuclei increases. Here you can see there are 5 groups. In group 1, there is only one lobed nucleus and their number is less, that is only 5 to 10%. In group 2, the nucleus is 2 lobed and their number is 25 to 30%. Group 3 is very important and the most active of neutrophils, which contain 3 lobed nucleus. Their number is 45 to 47%. They are maximum in number and they are the most active neutrophils present in our blood. Group 4 has 4 lobed nucleus. Their percent is 16 to 18%, and group 5 is the group which include 5 or more lobes in nucleus, and they are minimum in number. Now here in this picture you can see all these groups. Group 1 5 to 10%, group 2 with 2 lobe nucleus 20 to 30%, group 3 with 3 lobe nucleus 45 to 47%, group 4 with 4 lobe nucleus, this is 16 to 18%, and group 5 with 5 to 7 lobes in the nucleus is just 2%. Now, as I just mentioned that the neutrophils with 3 lobed nuclei are most abundant, they are fully mature and most active neutrophils. Now, just we have seen in the picture from left to right, the number of lobes increases. So, presence of younger neutrophils, that is with less number of lobes in the nucleus, that is shift to left, that means towards less number of nucleus, indicates stimulation in bone marrow. That means the bone marrow is producing fresh neutrophils in more number. Then reverse to it, if there is a shift to right, that means if there is a shift from lesser number of lobes to more number of lobes in the blood, then it indicates that there is some kind of suppression in the production of neutrophils in the bone marrow. So, it indicates suppression of bone marrow. Now, neutrophils are phagocytic in nature. They provide defense mechanism to our body by phagocytosis. That means they engulf the foreign pathogens or other materials into their cell body and digest them with the help of certain enzymes and chemicals. Mostly they contain bactericidal and antimicrobial chemicals, especially enzymes in their granules, which are helpful to destroy the microorganisms. They ingest them, digest them with the enzymes, and then exclude the remaining part. So that completes the process of phagocytosis in the neutrophils. Now, neutrophils provide non-specific defense against pyogenic pathogens. As I just mentioned, pyogenic pathogens are those when some body part is infected with such pathogens, then there is an inflammation in that part. The WBCs, they are accumulated, extra fluid accumulated, the area becomes red and ultimately pus is formed. In such cases, neutrophils provide a non-specific defense. When we talk about immunity, we always talk about immunity is broadly categorized. Body defense mechanism is categorized broadly into two categories. One is specific defense and one is non-specific defense. When we talk about non-specific defense, that means any type of pathogen when enters and when its composition, especially protein, which is otherwise called as antigen, is different from our body protein, then there are certain mechanisms, there are certain cells which identify that this antigen, this protein does not belong to our body, and they just engulf it and digest it by phagocytosis. So this type of defense is called non-specific defense, whereas in case of specific defense, a specific antibody is produced against a specific pathogen, maybe bacteria or virus, and it can only destroy that type of pathogen. Now, as I just mentioned, they provide first line defense against acute bacterial infection. That means whenever there is an infection which causes illness, then if it is of bacterial nature, then the neutrophil count increases and these neutrophils by phagocytosis destroy them. So it is a first line defense. So whenever any pathogen enters in our body, it has to encounter with the neutrophils first. So they are the first and front line defense cells of our body. Now, in inflammation caused by bacteria, neutrophils move to the site of infection by chemotaxis. Now, what happens? When there is an inflammation, certain chemicals are released from the body cells, from the pathogen, and from the inflammatory condition. Those chemicals attract the neutrophils towards the site of infection, and this phenomena is called chemotaxis. Now, another phenomena by which the neutrophil reaches to the site of infection and they are gathered there, they are accumulated there, is the process called diapedesis. When the blood passes through the capillaries capillary is like this, having minute holes in the capillary wall. Now, when WBC is here, neutrophil. Now, it will be attracted by chemotaxis. So, chemotaxis, now what will happen? This WBC will form a small pseudopodia here and try to escape from this minute hole of the capillary onto the site of infection, and gradually this pseudopodia increases and the WBC reaches on the site of infection. This special phenomena is called as dipedasis. Now, coming on to the eosinophils. Eosinophils are the leukocytes which are responsible for protecting us from the allergies and allergic substances. Like when there are some skin infections, maybe fungal infection, maybe bacterial infection, or one may have bronchial asthma. Then in case of hay fever, in case of parasitic infections, where there are some intestinal parasites or other parasites in our body, they secrete certain chemicals which are giving allergy. So, in those conditions, eosinophils have the work to do. The eosinophils are found more in tissue as compared to blood, because their main function is in the tissue. So, therefore, their ratio in tissue to blood is 100:1. That is 100 eosinophils in tissue and only one part in the circulating blood. Now, eosinophil, as I just mentioned, are the leukocytes which are granulocytes and take the stain by a basic dye called eosin. It is a red colored dye, as you can see here. The nucleus of eosinophil is bilobed. They are about 10 to 14 μm in diameter. Their count is 2 to 4% in differential count, or their total count can be 40 to 440 per cubic millimeter of blood. Nucleus, as I just mentioned, the eosin, as I just mentioned that they take up eosin. Then important is they are amoeboid in nature. As we have seen just now, the neutrophils, by being amoeboid in nature, they form pseudopodia and they comes out from the blood capillaries into the affected part of the body and perform their function in defense mechanism. The similar are the eosinophils. Now, coming on to the functions of eosinophils, as I just mentioned, in case of neutrophils, eosinophils are also attracted towards the site of infection with the help of chemoattractants secreted by the mast cells which are present in the tissue. Then eosinophil attacks large parasites which cannot be engulfed by phagocytosis by the neutrophils and monocytes. These specific granules of eosinophil, they contain eosinophil cationic protein, that is ECP, then eosinophil peroxidase, that is EPO, and eosinophil derived neurotoxin, that is EDN. These toxic proteins stimulates the mast cells to produce more histamine and helps to attract more eosinophils to the site of infection. You know, the mast cell secretes HHS, that is heparin, histamine, and 5-hydroxytryptamine or serotonin. So, these stimulates the mast cells to release more histamine, and that is how more number of eosinophils are attracted towards the site of infection. Now, eosinophils attack the intestinal helminthic parasites, especially Physiola hepatica, roundworms which are present, hookworms. They may attack the larval stage of parasites. You know, the toxic proteins of eosinophils especially attack the larval stage of the parasite, which are more susceptible to these chemicals. Then eosinophilia, as I just mentioned, philia means more. When the count of eosinophils increases, it is called eosinophilia. It occurs in parasitic infections. It occurs in allergic conditions of skin, in hay fever, in allergic bronchial asthma. As you know, some people are very allergic to pollen grains, and when they especially passes in that season from an area where a lot of flowers are there, lot of plants are there, they get an allergic reaction which is called as allergic bronchial asthma. Then Eocinopenia is the condition in which the number of eosinophils decreases. It is seen in certain conditions like glucocorticoid therapy, when glucocorticoid hormones are given for the treatment of certain diseases, or in case of aplastic anemia, that means when the bone marrow does not work properly to produce blood cells, that is called aplastic anemia, and it is in case of drug induced agranulocytosis. Now, basophils. Basophils are the neutrophils, the leukocytes, granular leukocytes, which are found in the least number. They are only 0.5%. They are smaller in size and they measure about 10 to 14 mu in diameter. Nucleus is slightly bilobed. It is kidney shaped. Then cytoplasm contain large granules which are stained with basophilic dyes. Granules are covered with membranes and contain dense particles called cracod-laden particles. Here is the picture of basophil having a bilobed nucleus and the granules in it, cracod-laden granules. Now, the functions of basophils. Basophils are mainly related to the allergic reactions. So when there is an allergy of some type to some substance, to some allergens, then the number of basophils increases. They act similar to mast cells, which are found in the tissue. Their granules contain all the three substances which are secreted by mast cell: histamine, heparin, and 5-hydroxytryptamine. Now, the antibody responsible for allergic reactions in our body is IgE. Now, this antibody attaches to the basophil and mast cells, and they rupture to release large quantity of all these three: histamine, heparin, and serotonin, commonly called as HHS, and with them bradykinin and lysosomal enzymes, and that is how they work against the allergic reactions. Now, in these three, that is HHS, the histamine is a vasodilator. So whenever there is an allergic reaction, vasodilation occurs and lot of blood is supplied to that part of the body where allergy occurs. So if there is a massive release of histamine, this causes a condition called as anaphylaxis, and it can result in a severe anaphylactic stroke. So in that case, you must have learned that in case of certain injections, certain medicines, a test is done prior to administrating that drug. One of that is penicillin, which is very common drug, antibiotic, and another is xylocaine, which is used as a local anesthetic agent, especially in case of dentistry. It is used for when there is some procedure is to be done. So just to block the nerve impulse conduction from that part, xylocaine is given to block the nerve impulse so that the patient does not feel pain. But it is highly reactive, and it causes anaphylaxis due to vasodilation. The basophils and mast cells, they play a very important role in resistance of host to some viral and bacterial, especially in case of parasitic infections. So when their number is increased, that indicates that some kind of infection is there. Their number increases in cases of smallpox, chickenpox, and tuberculosis, commonly called TB. Now coming on to granular leukocytes, that means the cytoplasm does not contain granules. After staining, they can be of two types. One is monocyte and another is lymphocyte. Now monocytes, the diameter of monocyte is about 10 to 18 μm. Their nucleus is large, which occupies almost about 50% of the cell. That means 50% cytoplasm and 50% nucleus. And sometimes when the monocyte matures and becomes larger, then this proportion becomes even more. This nucleocytoplasmic ratio becomes more as the size of nucleus is bigger and it is displaced to one side of the cell. Nucleus is round in young cells but becomes kidney shaped or horseshoe shaped as the monocytes mature. Now here is the structure of a monocyte. It has got a very large nucleus which is bilobed or kidney shaped. You know, the half-life time of monocyte in circulating fluid, circulating blood is 10 to 72 hours only. But when they enter into the tissue system, they are converted into macrophages, tissue macrophages, and then they can live for weeks or few months, and they can perform their function as macrophages. Then the normal monocyte count in blood is only 2 to 8% of leukocytes, and in the blood their count is about 1500 to 4000 per cubic millimeter of blood. Now neonates have more number of monocytes as compared to adults, that is about 10,000 per cubic millimeter. Now there is a difference in the monocyte count in male and female. So in case of man, the number of monocytes is more as compared to women. Now again, the term monocytosis and monocytopenia. Monocytosis is a term which is used for increased number of monocytes in blood as compared to the normal number. Now, if the number increases more than 8,000, it is called monocytosis. It is reported in certain diseases like syphilis, one of the sexually transmitted diseases, then tuberculosis, a respiratory disease, then leukemia, kind of blood cancer, and in certain viral infections. So the number of monocyte increases to cope up with that kind of infection because the monocytes are phagocytic in nature. Now monocytopenia, that means decrease in monocyte count. It is seen in case of hypoplastic bone marrow. That means the formation of monocyte series CFUM is not proper in the bone marrow. So the number of monocytes will decrease in number, that is called as monocytopenia. Now coming on to the second agranular leukocyte, that is lymphocyte. Lymphocytes are heterogeneous group of leukocytes. Some are larger ones, some are smaller ones. Their morphology is variable, therefore they are called as heterogeneous group. These are grouped into two categories. One is the small and other large lymphocytes. In fact, the small lymphocytes are formed from large lymphocytes. Now, the characteristics of a small lymphocyte, they are smaller in size, that is about 6 to 9 μm in diameter. These are formed from the larger lymphocytes. They constitute about 35%, that is 20 to 50% of lymphocytes. Then their cytoplasm is bluish with some smaller granules. Nucleus is ovoid. It is kidney shaped, which is usually centrally placed. That means nucleus is placed in the center of the cell and it occupies 90% area of the cell. We just mentioned that the nucleocytoplasmic ratio is more, but here you can see as the cells becomes smaller, the size of the nucleus in respect to the cytoplasm becomes more, and that is 90%. Now, larger lymphocytes, they are 10 to 15 mu in diameter. They are fairly mature cells and they do not divide any further. They constitute about 60% of the total lymphocytes, and they contain very small pink granules. In such case, when such granules are found, they are called NK cells or T cells, that is natural killer lymphocytes or T lymphocytes. Their nucleus is oval and it is displaced towards one side of the cell. We call it eccentric nucleus. Eccentric nucleus. Now coming on to the lymphocyte count, the normal lymphocyte count is 500 to 8000 per cubic millimeter of blood. Normal lymphocyte count is about 20 to 40% of the total leukocytes. If the count of lymphocyte increases, then the phenomena is called lymphocytosis, and when the count of lymphocyte decreases than the normal, then the phenomena is called as lymphopenia. Now, functionally, the lymphocytes can be grouped into three categories, that is B cells, T cells and NK cells, that is natural killer cells. Now, B cells are basically responsible for providing humoral antibody, that is antibody mediated immunity. The T cells, which are the cells which matures in thymus, they mediate cellular and cell mediated immunity. Some of the T cells are also called memory T cells. They retain the memory for the immunity for the future. Now, another cell is T helper cell, which produce cytokines on contact with pathogen for killing it. Another type of cell is the cytotoxic T cells, which produce toxic granules which contain enzymes which kill the infected cells. So that is how these three types of lymphocytes contributes their role in the immunity. Now, type of lymphocytes. Normally, the lymphocytes are categorized into two categories, B lymphocytes and T lymphocytes. These B and T lymphocytes generate specific response to specific pathogens. When one is infected by a specific pathogen, a specific immunity is developed with the help of B lymphocytes and T lymphocytes. T lymphocytes are those which are matured in thymus and B lymphocytes are those which are produced in bone marrow and released in the blood. Now, B and T cells gather sensitization memory against a specific pathogen and form memory cells. So, one type of lymphocyte, T lymphocyte, they are called memory T lymphocytes. Now, when first time a person is infected with a specific pathogen, a specific antibody is produced against that particular pathogen, and the cell memorizes how to produce that protein of antibody, and those cells which such a memory are stored in the body in lymphoid organs for a long period, and whenever that person is infected by the same pathogen next time, then within no time large number of antibodies are produced. So, those cells are called as memory T cells. Now, another category is natural killer cells. These are also lymphocytes. They mediate natural and non-specific immunity. As we have seen in the case of neutrophils and monocytes, they are important for providing non-specific immunity against the pathogen. Here NK cells, these are natural killer cells. They provide cytotoxic innate immunity. When we say innate immunity, means since birth. And they defend the host from tumors, tumoric cells which are to be destroyed, and the cells which are infected by virus, they are to be destroyed. So, they are identified and then destroyed with the help of natural killer cells. Now, let us summarize the functions of leukocytes or white blood cells. One of the important and foremost function of the leukocyte is the phenomena of phagocytosis, by which the neutrophils and monocytes, they engulf the bacteria and foreign particles, digest them. Monocytes kill tumor cells and sensitization of lymphocytes, they are also instrumental in that. Then second function is the antibody formation. As I just explained that the B lymphocytes are instrumental in production of antibody. They are further differentiated in the tissue as mast cells, and mast cells produces antibody for a specific pathogen. So, this is a specific humoral immunity. Now, they prevent blood clotting. The basophils secrete a chemical called heparin, which is important in preventing intravascular clotting. You know, whenever there is a damage to blood capillary or blood vessel, even inside the body, so there is a tendency of blood to form a clot. And when the clot is formed inside the circulatory system, it is called as thrombus. So, the heparin prevents formation of clot inside the circulatory system. Then, monocytes produces prostaglandins E and clot promoting factors. Now, the monocytes have got this property, they secrete prostaglandin E and clot promoting factors. Now, a condition when there is a abnormal decrease in the leukocyte count, then it is called as leukopenia. That is, decrease in the leukocyte count. It is commonly seen in viral infections. If we take example, HIV, AIDS, in case of protozoan parasite malaria, in case of typhoid, in case of aplastic anemia, the leukopenia occurs. Aplastic anemia means bone marrow does not form the blood cells properly. Then it is also seen after chemotherapy and radiotherapy. As you know, in case of malignancy, in case of cancer, the most common treatment is the radiation therapy and chemotherapy. In that case, the number of WBCs reduces. Even in the case of prolonged use of corticosteroid drugs, the leukopenia occurs. Then leukemia. Leukemia, just reverse to this, leukemia means abnormal increase in the number of blood cells. Leukemia is defined as malignant neoplasia of hemopoietic cells, means the hemopoietic cells which are responsible for production of blood cells, they increase their production and abnormal number of WBCs are produced. It is observed in case of tumors of bone marrow, because you know the formation of leukocytes, that is Leucopoises, occurs in bone marrow. So if there is a tumor in bone marrow, obviously large number of WBCs will be formed there. Now coming on to the types of leukemia. Now here is one, acute lymphoblastic leukemia called ALL. It is a primary disease of children and young adults. The internal organs are infiltrated with leukemic cells. Then acute myeloblastic leukemia, another type of leukemia, AML, it primarily occurs in middle age group. ALL occurs in children and young adults. Acute myeloblastic leukemia occurs in middle-aged group, 15 to 40 years. In peripheral blood, more than 60% leukocytes are in myeloblastic stage. That means the bone marrow is forming more and more number of stem cells or the mother cells responsible for forming leukocytes. We call them myeloblast cells, and in normal condition they do not come in the peripheral blood. But here, since their production is more, so these immature myeloblastic cells, rather than getting matured into leukocytes, different type of leukocyte, they enter into the circulation, and that condition is called acute myeloblastic anemia. Now coming on to the differential leukocyte count. Earlier we have seen that counting the leukocytes gives an elementary idea about the type of illness the patient is suffering from. But you know, as we have seen that all these leukocytes have different functions in different situations, in different physiological conditions and in different pathological conditions. So their differential count definitely indicates about the type of infection one is suffering from, so that the doctor can reach on a conclusion for deciding the treatment. The neutrophils are 2,500 to 8,000 per cubic millimeter. That is, they form the major share of leukocytes. that is 55 to 70%. Then comes eosinophil, which are about 50 to 500 per cubic millimeter, forms only 1 to 4% share of the leukocytes. Then among these granular leukocytes, basophil count is the minimum, that is only 0.5% to 1%, and their count in the blood ranges from 25 to 100 per cubic millimeter of blood. Now, amongst the agranular leukocytes, the lymphocyte contribute about 1000 to 4000 per cubic millimeter. It can increase or decrease depending upon the physiological condition. They contribute about 20 to 40% of leukocytes. Then monocytes, they are just 100 to 700 per cubic millimeter of blood and contributes only 2 to 8%. Now, for this differential leukocyte count, a blood film is made on the slide and the smear is stained with differential stains. So, the different type of cells take different stain, as we have already seen. The neutrophils take neutral dye stain, the eosinophil take acidic stain, the basophils take basic stains, and so on. So, then they are counted under the microscope, and that counting is called as DLC, or differential leukocyte count. Now, just for your reference, here is a picture that how these cells look under the microscope after staining. Here you can see the neutrophils with the polymorphic nuclei, they form 55 to 70%. The eosinophils with red stain of eosin acidic dye, they form 1 to 4%. They are basophils, looks like this, and only 5 to 1%. Then there are lymphocytes. These are small lymphocytes. These are large lymphocytes with bluish cytoplasm and larger nucleus. Then there are monocytes here with kidney-shaped nucleus. They are only 2 to 8%. Now, coming on to the method of total leukocyte count. As we have seen in case of RBC count, the same instrument used here also. It is called hemocytometer. In fact, this hemocytometer is used for counting of both, for counting of total number of RBC and for counting total number of WBC. So, here principles of this method is blood is diluted with the fluid which causes hemolysis of RBC, but WBC remains intact. Then these are counted in the Neubauer's chamber. Gentian violet, that is important because you know white blood cells are called so as they are almost colorless, and red blood cells are called so because they are red colored when we observe them under microscope. So, for counting WBC, some kind of fluid has to be used in which there is a color. So, gentian violet is used in the diluting fluid for counting leukocytes. Now, here you can see the pipette used for the purpose. There we have seen in the counting of RBC, the pipette was having a red bead inside, but here in this case you can see there is a white bead in the pipette. There is a mark here of 0.5 up to which the blood sample is taken. Then with the help of this suction tube, the diluting fluid is sucked in up to the 11 mark, and then it is continuously mixed, and first few drops of the sample are rejected because that part of the blood is not properly mixed with the diluting fluid. It is shaked for 1 minute, and then it is poured into the Neubauer's chamber for counting of WBC. Here you can see the same counting chamber which was used for counting of RBC. You know, the counting of RBC is done in this portion, whereas the counting of WBC is done in these 4 corners. As you can see, these 4 corners are meant for counting of WBC, and each corner has got 1, 2, 3, 4, that is 16 small squares. In all these, the white blood cells are counted, and then Y is the number of total WBCs counted into 5. 5 means the area of these chambers and depth of it, then dilution factor. So, Y into 50, that comes the total leukocyte count per cubic millimeter of blood. That's all for today. Thank you very much. In the next lecture, we will discuss blood groups and blood transfusion. Thank you.